In the 1950s, a group of ranchers in Idaho were baffled when their sheep gave birth to lambs with a singular deformity. Mystified by these Cyclops sheep, they called in scientists from the U.S. Department of Agriculture to investigate. The researchers hypothesized that the pregnant ewes had snacked on poisonous birth defect-causing plants. They collected the local flora and fed samples to lab rats, but struggled to replicate the effect. So they decided to directly observe the sheep, with one scientist even living with the herd for 3 summers. After a decade of trial and error, the scientists finally found the culprit: wild corn lilies. The lilies contained an active molecule with 6 connected rings that they named cyclopamine in reference to the Cyclops sheep. They didn't know exactly how cyclopamine caused the defect, but told ranchers to steer clear. It took about 4 decades before a team of biologists led by Professor Philip Beechey stumbled upon the answer. His lab was studying a specific gene found in many species, from mice to humans, called the hedgehog gene. It was named by 2 scientists who later shared the Nobel Prize for their work, who found that mutating this gene in fruit flies produced pointy spikes, like a hedgehog. Beechey and his colleagues performed genetic modifications to turn off the hedgehog genes in mice. This resulted in severe defects in the development of their brains, organs, and eyes, or rather, eye. Then, while perusing a textbook, Beechey came across photos of the Cyclops sheep and realized what had eluded scientists for 4 decades. Something must have gone awry involving the hedgehog gene. Let's take a step back. Genes contain instructions that tell cells what to do and when to do it, and they communicate their directives using proteins. The hedgehog gene itself tells cells to release a so-called hedgehog protein, which kicks off a complex series of cellular signals. Here's how it works in normal, healthy development. Hedgehog protein latches onto a protein called patched. That inhibits or holds patched back, allowing another protein called smoothened to freely signal the cells, telling them where to go and what kind of tissues to become. Cyclopamine, say in the form of a delicious corn lily, interrupts this pathway by binding onto smoothened. That locks smoothened up so that it's unable to send the signals needed to mold the brain into 2 hemispheres and form fingers or separate eyes. So even though the hedgehog protein is still doing its job of keeping the way clear for smoothened, cyclopamine blocks smoothened from passing along its chemical message. That settled the science behind the one-eyed sheep, but Beechey and his team caught the glimmer of another, more beneficial connection. They noted that uncontrolled activation of the smoothened protein was associated with a human syndrome. It's known as basal cell nevus syndrome, and it predisposes people to certain cancers. The scientists proposed putting cyclopamine's smoothened binding powers to good use as a treatment for these cancers, as long as the patient wasn't pregnant. Unfortunately, researchers eventually found that cyclopamine causes negative side effects, and its chemical properties make it difficult to work with. But they did discover that closely related molecules are safe and effective, and 2 of these drugs were approved in 2012 and 2015 as skin cancer medicines. When those farmers first saw the Cyclops sheep, they could have chalked it up to a freak genetic mutation and walked away. Instead, their decision to investigate turned a mystery into medicine, showing that sometimes there's more than meets the eye. TED-Ed is a nonprofit. If you value our work, please consider supporting it on patreon.com/TED-Ed.